Long ago, as humans were developing, B cells stepped up to become the valiant warriors of our internal world. Their mission? To bravely fight against countless foreign invaders. And now, our story begins. Deep within our bones lies a bustling city called the bone marrow. Within this vital organ reside special cells known as hematopoietic stem cells, or HSCs. Our HSC embarks on a specific path, eventually transforming into what we call a common lymphoid progenitor. In the transition of a common lymphoid progenitor to an early pro-B cell, the recombination activating genes 1 and 2 act as the genetic scissors that initiate the rearrangement of the heavy chain genes. Through the expression of recombination activating genes, it transitions into an early pro-B cell. Here, the real magic begins. Specific segments of its DNA, known as variable V, diversity D, and joining J gene segments, started to get rearranged. There's a crucial rule called allelic exclusion. This means that the first chromosome to successfully complete its D and J gene segment splicing will signal the other chromosomes to stop trying. During allelic exclusion, the heavy chain D and J gene segments of the first chromosome splice together, forming the late pro-B cells. Now, the late pro-B cell takes the successfully spliced DJ gene segment, attaches a V gene segment to it. This newly formed heavy chain gene then gets linked to the Mu gene, which is the key to developing the antibody component. The Mu gene specifically codes for the constant region of the IgM antibody. So, at the pre-B cell stage, our developing B cell starts producing its very first type of heavy chain, the foundation for IgM antibodies. After completing light chain rearrangement, the developing B cell expresses a functional B cell receptor, forming an IgM plus a mature B cell. These cells are tested for self-reactivity through a process known as central tolerance, regulated by the autoimmune regulator IRA. If the B cell binds strongly to self-antigens, it undergoes apoptosis. If binding is intermediate, the cell gets another chance by editing its light chain gene switching between kappa and lambda loci. The selection occurs via one of two mechanisms: positive mechanisms and negative mechanisms. Positive mechanism occur via antigen-independent signaling, where if the receptors on the B cells do not bind to their ligands, the cells do not receive proper signals and cease to develop. A negative selection mechanism occurs by the binding of self-antigen to the beaker, where if the beaker can bind strongly to a self-antigen, the development of B cells is ceased. After successful negative selection, immature B cells exit the bone marrow and enter the spleen, where they become transitional B cells and begin expressing IgD alongside IgM. The cells migrate to lymphoid organs as they tend to have a constant supply of antigen with the help of the circulating lymph. The migration is induced by chemokine interaction between CXCL13 and CXCR5. The mature B cell carries both IgM and IgD and patrols lymphoid tissues in search of its specific antigen. When the B cell encounters its matching antigen and receives help from a T helper cell, it becomes activated and begins clonal expansion. B cells can be activated in two ways: T cell dependent and T cell independent. At the beginning of the T dependent B cell response, the B cells bind to the antigen via the Ig receptors. Some of the antigens are internalized into specialized vesicles within the B cells. The internalized antigens are processed and re-expressed in the form of peptides, presented in the antigen binding groove of class II MCHE molecules. T cells that have been previously exposed to antigen-bearing dendritic cells can now bind to the MCHE presented peptide on the surface of the B cell. The binding is further enhanced by the interaction of accessory molecules on the T and B cell surfaces. Some of the T cell activated B cells now move into specialized regions of the lymph node or spleen to begin the process of Differentiation. Some B cells can be activated without help from T cells. This is known as the T cell independent B cell response. It happens when B cells encounter special antigens called Ti antigens, which are usually large molecules with repeating patterns found on microbes. This response is fast, allowing the immune system to react quickly, but the antibodies produced are usually lower in affinity and less diverse compared to those from cell-dependent responses after activation by antigen and T cell signals. Antibody-producing plasma cells were entered germinal centers to undergo mutation and class switching. Some become long-lived plasma cells, while others become memory B cells that stay in the body for faster future responses.